Hello, good morning. This is the third video to accompany the third PowerPoint presentation on the VLE. Just made a few changes again, so I'll be updating the VLE after this recording. So let's have a look at the brief again. And again, a reminder, 3,500 words, okay, as a booklet for overseas health professionals. Now, in PowerPoint 1, we covered assessment criteria 1.1, 1.2, PowerPoint 2 and video 2. I covered disease transmission through vectors 2.1 and the non-specific defences that the body uses to combat infection. So we're on to assessment criteria 3.1 with that one. And now in the third video and PowerPoint, we're having a look at assessment criteria 5.1, 4.2 and 4.3. Probably just the first two, because we don't want to make it too onerous. Okay, so remember your own work. It's the first time in this video and handouts that I have used a Wikipedia, because the image that I've referenced is applicable on antibodies. Okay, so I'm going to stop sharing that one and I'm going to start to share the PowerPoint. As I say, just made some changes earlier this morning, so I will ensure that I put that on the VLE, the new version, shortly. And here we are. So use of drugs to fight infectious diseases, humoral and cell-mediated immunity. We'll see if we can get through to 4.3. Okay. And okay, so just adjusting picture on my screen to the top right. So antibiotics, antiviral and antifungal drugs. Okay, now because these drugs are actually trying to fight off infections from bacteria, that's antibiotics, antivirals and antifungal drugs. Each of them are attacking particular structures on these types of organisms. And because antibiotics are no good against viruses, that's because viruses have different structure and so do fungal cells. So what we need to think about is what drug we need to use, and is it appropriate for the microorganism that's infecting the host. Okay, so there's a vast number of medications within these antibiotics, antiviral and antifungal drugs. Unfortunately for antibiotics, there's very little research done on new antibiotics because the costs don't reap, unfortunately, the profits that pharmaceutical industry requires. And so research into that started to wane in the 1980s and 90s. But then when we saw multiple resistant antibiotics, that research has now taken on more serious overtones, because we are running out of new classes of antibiotics. In fact, a new class of antibiotic hasn't been discovered for 20 years or more. And again, this is all down to costs of research, etc. and the amount of money that can be returned. So vast amount of medications for the treatment of infectious diseases. Some of these medications are what we call broad spectrum drugs or antibiotics. So a broad spectrum drug means that it can be used for a number of infections, but some are very highly specific. And so we target various drugs to particular infections. And prescribing a drug has to take into account the following. Is this the best drug to use for the patient? Are these alternatives to the one chosen? What other medication is the patient on? And will there be any interactions? And what is the cost? Now, for someone who's working overseas, you've got to remember that some countries are not particularly affluent. Their health services are not geared up for high-cost interventions. And so a cost can be a contributing factor to these countries and an overseas worker. So point this out in your booklet. So cost is going to be a consideration. So antibiotics work by disrupting certain aspects of the bacterial cell wall. Now remember, they're eukaryotic cells. Unlike us, we have just a cell membrane. They have cell membranes and an extra layer, the cell wall. And it's that some of the drugs, particularly some of the antibiotics, as penicillin is the example here, disrupts the cell wall. So as the penicillin, which is when the penicillin, is used, it actually stops the bacterial cell wall from actually dividing. So when the bacteria grows and divides by binary fission, okay, what it does is it stops the bacteria from remaking a cell wall, so the bacteria dies because it's not enclosed, and so all the cell contents just spew out into the environment and it dies. Okay, so it stops the bacterium from multiplying, and as it grows, that cell wall material is blocked. Now the substance is called peptidoglycans, and that supports the overall cell structure, and holes in the wall occur because the penicillin has stopped the formation, and holes appear in the cell wall, and that causes water just to flood into the cell, causes the cell to burst, and the bacteria dies. Okay. Now there are some other types of disruption that carries on. For example, the nucleic acid synthesis. Remember, these are things like the DNA and the RNA within the nucleus. So this particular drug, cycloprofloxacin, is an antibiotic, works by inhibiting two specific enzymes used in the making of DNA, what we know as DNA replication. And remember, DNA is deoxyribonucleic acid. Now, this causes the DNA to fragment into pieces and prevents the bacteria from carrying out cell division. Remember, DNA, it's in a long chain in a bacterium and coiled round. And what happens is the enzyme splits up the DNA, so there's no continual code to code for a new bacterium, and so therefore again the bacteria dies. Now another type of way that the antibiotics work is what we call protein synthesis. So this is targeting the RNAs. Tetracycline this drug interferes with protein that's essential for the bacteria's growth and so results in the cell's death. So that's 3 ways in which antibiotics actually work. Disruption of the cell wall, causing holes to appear and water flooding in, bursting the cell. Disruption of the DNA, so it can't code for important compounds, or disruption of protein synthesis, so proteins can't be made and results in the bacterium's death. Now, with antiviral medications, antibiotics won't work because they've got a totally different structure and should never really be described. This is one of the problems we've had with medics and other health professionals, including dentists, etc., prescribing antibiotics when they weren't necessary. Now, it's not just limited to health professions that in humans. Veterinarians also prescribe antibiotics to animals. Now, if those animals are in our food chain, so for example, sheep and pigs and cattle, etc., and chickens, then some of those antibiotics still persist in the meat. We're exposed to it, and again, that can lead to resistance because of our kind of like exposure to them. But antiviral drugs can also be a problem in food chains. So we have to be careful about prescribing them. So the reason that the virus antiviral medication is different is because it doesn't possess cell wall nor ribosomes. So we can't disrupt the cell wall. So that's why antibiotics are useless. And it has no ribosomes, those little structures within the cell that are responsible for protein synthesis. And therefore, the antiviral drugs have to look at a different mechanism to disrupt the cell. And in this case, all right, the treatment for shingles is an antiviral drug called a cyclovir. So a cyclovir that inhibits certain stages in the viral replication. So it's interfering with the process of the virus trying to assemble itself within your cell. So antiviral drugs such as this, that's how they actually work. Antifungal drugs, amphotericin, along with other antiviral drugs, inhibit the production of new proteins for the production of their cell wall. But again, structurally slightly different to the bacterial cell wall. So again, we have to have a different drug to look at different proteins to inhibit. When we come on to anti-parasitic medication, so for example, that of protozoans, chloroquine is used to treat malaria. It's one of the reasons why the British were very famous for drinking gin and tonic in the tropics, because the tonic water contains quinine, and that's why it's slightly bitter. And it comes from a plant source, and it's put into the drinks, and it gave you some anti-malarial protection from this protozoan. But chloroquine is what we actually use as a drug, but there are others such as mebendazole and albendazole, used for worm infections such as the Ascaris lumbricoides. So there we've got a protozoan drug, chloroquine, and 2 drugs that do the treatment of worm infections, Ascaris lumbricoides, the intestinal worms. Okay, so again, I would mention all these in your booklet. So how does drug resistance arise? Well, in the last 30 years, due to increased use and misuse of these antibiotics, we found a rise in antibiotic resistance. So scientists are now trying to find unusual places where antibiotics exist. And some are looking at sea creatures that produce antibiotics because they're in an aquatic environment and continually exposed to various bacteria, etc. And so therefore we're kind of like trying to look to see if they could be harnessed as new classes of antibiotics, because we're coming to the point where we're having serious infections that we can't treat. So unfortunately, the misuse of antibiotics has given the potential for bacteria to change and mutate. Now, remember, mutation is kind of like just a change in the outside coating of the bacteria. So it's like me here, you recognize me because you can see, and I know this looks going to look rather silly, but you recognize me with my face. But if I do this, change my outside appearance, okay, looks a bit funny on the tube, but if I'm like this, you think, is that Frank? Is it not? Okay, and that's exactly what the bacteria has done. It's changed its outer coat. Okay, sorry about that, but it's a useful illustration. Okay, because the body goes, oh, I'm not sure, is that salmonella? Is that tuberculosis bacterium? Because it's changed its chemical nature, and they mutate and they can survive and they can reproduce, passing on this resistance in a small piece of circular DNA within the bacterial cell known as a plasmid. Many, many years ago, although scientists recognized this little circular disc of DNA, which they termed a plasmid, they didn't think it was particularly important. But then they slowly realized that it actually is what confers resistance. It's like a little memory chip that the bacteria uses to remember it's been exposed to a particular antibiotic, and it can pass that between bacteriums. So, of course, the most famous of the types of mutations that we see in bacteria are highlighted by the bacterium Staphylococcus aureus. Now, I know it sounds like a bit of a mouthful, but you will recognize it lower down in a moment. Now, this has become resistant to one of our most powerful antibiotics known as methicillin. And Staph aureus, we can truncate the whole Staphylococcus bit into Staph aureus, is a common bacterium found on human skin. It's on your skin, no problem. Remember, we talked about the skin being an impermeable barrier as long as it's not cut and it's kept intact. And it's normally harmless and cause little or no harm. However, if you cut yourself, that bacterium has the opportunity to get into your blood and cause an infection. Now, in the past, we would have used ordinary antibiotics and they were fine. But then, because of the overexposure over numerous decades, this strain has mutated in what we know as methicillin-resistant Staphylococcus aureus, or MRSA. Most people have heard of MRSA. Now, this is a very serious infection, because once it's got inside, it's like being let loose into a chocolate factory. The virus just thinks, whoopee, hit the gold mine. I can now grow uncontrollably, and there's nothing you can do about it. Even your most powerful antibiotic is useless against it. So this is why we need to swab the area. So health workers here, all aboard, if they're considering surgery, they have to look at swabbing the area where the surgeon's going to cut, or the health professional is going to cut. That's tested to see if they have MRSA. So you have to take special precautions during the surgery. Okay, there's only a handful of other medications we can use, and one of those is another antibiotic called vancomycin. Now, unfortunately, we are seeing the rise of VRSA, vancomycin resistant Staphylococcus aureus. And so this has a very serious implication for future control of infectious diseases. Because once we run out of these, we have nothing. And people could die from simple cuts, things like this. So we've got to be very, very diligent and the use of our antibiotics at the moment. Okay, and we move on. 4.2, now we're having a look at what we call humoral and cell-mediated immunity. So this is what can the body do. We've seen what the medics can do as far as antibacterial, antiviral, antifungal, antiprotozoic, anti-helminthic, which are the worm medications. We're now looking at what can the body do to defend itself when it's in the body. Now, the first one is the humoral, the antibody-mediated immunity. Now, this involves the production of antibodies against foreign antigens. So again, if I was a bacterium and this was a protein stuck on my cell wall, that would act as an antigen to trigger my body to say, oh, this isn't part of me, I need to fight this off. So the antibodies are produced by a type of lymphocyte known as the B lymphocyte, or B cells. Mature B cells are stimulated into secreting antibodies, and these mature B cells are also called plasma cells. Now, these antibodies can be found in the body's extracellular fluids, such as blood, plasma, lymph, in your lymph system, and the mucus, etc. And also, these antibodies are on the surface of the B cells, and they help to defend the body against bacteria, bacterial toxins, and in some cases, viruses. Remember, these are antibodies, not antibiotics, all right? Don't confuse the two, okay? Antibodies are used by the body to defend against viruses. Now, they circulate freely in the bodily fluid, so in your blood and in your plasma, before they enter the body cells. Okay, they can also cause the organ transplant rejection, because the body rejects any foreign object or organ. So if you donate a kidney or a heart or a liver, that's why we have to tissue match so very, very carefully. We also have to give the patient immunosuppressants, because the body is thinking, is that mine? Isn't it? Do I attack it? Don't I? So what we have to do is give it immunosuppressant, the patient immunosuppressant drugs, so that they can not reject that particular organ transplant. So antibodies produced by the B lymphocytes, these B cells, They're produced by mature B cells that secrete the antibodies, and they're called plasma cells. The antibodies are released into extracellular fluid, which consists of the blood, the plasma, the lymph, and mucus, etc. And they're also present on the B cells, and they defend against these bacteria and viruses that might have invaded our body. Okay. So production of lymphocytes in your bone marrow, or when you're in a fetus, in your mum's womb, in the fecal liver, we have these stem cells. Now, we've talked about stem cells. Remember, they haven't been given a particular role. So the technical name is called a pluripotent stem cell. That means it could go off to be any type of cell. Right, everything from a red blood cell to a liver cell, to a skin cell, to an eye cell. So it can be turned into any. Now, in this diagram, we're seeing that it's being turned into a lymphocyte stem cell. And these can be stored within the bone marrow, but also the thymus. Now, if it's stored in the bone, then we have B cells. And it's one way of remembering it's because it's originated in the bone and stored there. Whereas those in the thymus are known as T cells, T for thymus. So the thymus cell actually stores T cells. Now, both are then circulated in the blood. They can be stored in lymph nodes. You have lymph nodes up here in your neck, underneath your arm, in the breast, in the groin. Those are the areas of lymph nodes. And if those swell, there's usually a serious infection, but it can also mean that there might be the start of lymphatic cancers, which we have to be concerned about. So any lumps like that need to be investigated. They can circulate in the lymph, as I say, and then passed into the blood. So the structure of an antibody, do not get panicked at this. They look like a Y shape. Now here we have letters V and C. Now C stands for constant, means that these do not vary. So they are the protein structure that remains constant for the antibody, but this part of the protein structure, the V sections, are either ends of the antibody structure. Those are the variable ends, and they will change, and they will change due to the infection. So chemically, its structure will change for the antigen on a Salmonella bacteria or a Clostridium bacteria, also for various viruses. So the antibodies that the body produces for the COVID virus, that V area will be modified by the body to produce antibodies specific for the antigen on the COVID virus and antibodies for the flu, all right, or any other virus. So it's responding to specific antigens, specific proteins on that organism's outer coating, whether it's a cell wall or a virus membrane. And the reason why is because the antigen here, you can see in the purple and the green, corresponding to these purple and green areas on the V sections here, they fit the antigen, okay, here, all right, on our virus or on our bacteria. And it's that it locks on. Once it locks on to the virus or the bacteria, that sends a flag to the cells of the immune system to destroy those viral or bacterial particles that have invaded our body. So what they do is these antibodies are acting as flags to the body to say, hey, over here, this is a foreign body you need to kind of like destroy. Okay, and it triggers that response, as we'll see later in this PowerPoint. So again, this is the Wikipedia image that I've shown. Here you can see the variable sections at the ends here in orange, and you can see that this particular antigen, which would be present on a bacteria or virus, matches the shape of those antigen binding sites on the antibody. And so if you had a blue antigen, I know it's a color, but if you had different protein antigen that we've color coded blue here, you can see the shape is totally different to the orange antigen. And so that shape of protein, you will need a different shaped antigen binding site. Okay. Now, within antibodies, also known as immunoglobulins by scientists, there are different classes, and they can be found in various parts of the body and have different roles. We don't have to get into that much detail, but I've listed these immunoglobulins, and they're given letters. They're actually given letters. And we have immunoglobulin A, IgA, immunoglobulin D, IgD, IgE, IgG, and IgM. Okay, so again, those are different classes of antibodies, stroke immunoglobulins. Okay. Now, the other type of immunity is cell-mediated. Now, this involves the T lymphocytes, all right, the ones that were in the thymus, the T for the thymus, that's where they're stored. Now, these recognize intracellular antigens on the surface of cells and organisms or tissues infected by viruses, bacteria, or parasites. So this is when the bacteria, the virus, or the parasite is actually inside your cell. Now, what happens is your cell will actually take the antigen off the invading bacteria that's within the cell and place it on the outside of its own human body cell, so your cells, to kind of like act as like a beacon. And the beacon's kind of like going, I'm infected, I'm infected, I'm infected. I know technically it's not shouting this, but chemically that's what it's doing. And so this antigen on the surface of our cells alerts our T lymphocytes, our T cells. Now, they bind to the antigen, activating receptors on it called cytokine receptors. Now, a type of T cell known as a helper T cell produces cytokines, okay, that cause the activation of T cells to develop what we call into cytotoxic T cells. Now, cytotoxic means that it was actually going to kill your cells, that's your cells, because in doing so, by killing your cell that's infected, it destroys also and gives the body the chance to destroy the virus or the bacteria within your infected cells. It's as if your own body cells are committing suicide, because for the greater good of the body, it actually sacrifices itself so that the rest of the cells don't become infected. So the helper T cell produces cytokines that cause the activation of T cells to develop into cytotoxic cells. And they will target the infected body cells, and they will lyse them. That's the technical word. They'll break them open, and that infected cell is destroyed. At the same time, the cytokinins influence B cells to produce those plasma cells that will produce antibodies. Okay, and also the cytokinins activate macrophages that engulf and devour. Those are the ones that spread round, engulf and take the bacteria into the cell, and they will digest them. And that's cell mediated. Now, continuing with cell mediated, the body also produces memory cells. Now, these are really important, okay, because B cells can be stored as B memory cells, while in the cell mediated response, the T cells can produce T helper memory cells and cytotoxic T memory cells. This is going to be summarized in a diagram in a moment. Now, these are extremely important because once you've been exposed to a particular bacterium or viruses, those cells, those memory cells, so the B memory cells, the helper memory cells, and the cytotoxic cells go, hang on a minute, I've come across you before. And that means that it has a blueprint already made. It's like getting out a plan, a map, and goes, oh, right, how did I make that particular antibody, or how did I deal with that particular bacteria or viruses? Because it has a blueprint, it will kind of like remember that it's been infected before. And that's the principle behind vaccinations. So after the primary immune response, when you're initially infected, the body recovers, hopefully, but if the body is exposed again in a second exposure, we all know that we've had a cold, and we get over it quite easily if we have a cold, because our body kind of like will make antibodies because it's been exposed by that particular cold before. But in the second one, all right, it will produce antibodies much more quickly because of the numerous memory cells that it has, the T and the B memory cells, and in large amounts to quickly control the disease before it gets a foothold. So here's a diagram categorizing the two. Now, this is a very useful diagram, and there's a copy reference down at the bottom, but please, if you need to use this, please do so. It summarizes the in a bit more detail, the humoral, that's the antibody mediated, and the cell mediated. So antibodies, they produce antibodies that go out into the blood. Cell mediated is the T cells actually attacking the virus or bacteria microbe. So humoral, we've got these antigens circulating in our blood from the bacterium or the virus. The B cell binds to the antigen for which it's specific. So here's our antibodies on the B cell surface, and there's the antigen binding specifically to that antibody. The B cell is often stimulated by T helper cells, and it differentiates into, remember, a mature B cell, which is a plasma cell, and the plasma cell will proliferate, that means grow and reproduce, and produce lots and lots of antibodies against the antigen. Now, remember, the antigen is a word we switch between anything that's a foreign body. So it could be a bacterium, it could be a virus, it could be a parasite, it could be an organ transplant cell. The cell-mediated immune system, this control if we have a look, we have a T cell. That T cell can produce what we call a cytotoxic cell, because we have helper cells that stimulate the T cell to produce either cytokinins. The cytokinins will activate macrophages that will engulf and lyse the bacteria or virus, or cytotoxic cells can also be stimulated into memory cells, and also the cytotoxic cells. So the T cell, through the help of the helper T cell, the cytokinins will stimulate B cells, the cytokinins will activate macrophages, it will activate the T cells to become cytotoxic cells. Those cytotoxic cells can become memory cells. So there's lots of ways that our body can defend us. Here's another diagram explaining the same thing, and again it's from Pearson, so you can use this one as well, and divides it up. Here's our first exposure to our antigen, remember bacteria, virus, etc. And this can stimulate the B cell. The B cell matures into a plasma cell that secretes antibodies that defends against these pathogens and neutralizes the pathogens and will cause them to be destroyed. The B cell can become a B memory cell, which in turn, if you're reinfected, will become a plasma cell. If the antigen is taken in to one of our cells, it will present that antigen on the outside of of our cell and says, please, I need to be destroyed. And that stimulates the helper cell and the cytotoxic cells. Okay, and those cytotoxic cells become active, and they will produce, they will bind to the pathogen and destroy them by breaking open the cell. They'll also attack cancer cells, because the cancer cell is running rogue, it's one of your cells. So this is why your own defense mechanisms start to attack cancer cells. So it's one of the reasons that we know that we have cancer. Now those cytotoxic cells can become also memory cells, and they can be both the B memory cells and the cytotoxic memory cells can be stimulated by these memory cells. So slightly more, less complex diagram than the last one, but please use either. This one's pretty good for using it as the explanation. The little arrows with the plus means it stimulates, and the brown arrows means they give rise to these other cells. So the helper cells give rise to memory cells. B cells give rise to plasma cells and memory cells. Cytotoxic cells give rise to active toxic cells and memory cells. So again, useful diagram. Okay, so last part, 4.3, you will need to use this graph, all right? And again, this comes from the brief, so you can quote the brief from a sentence. Need to put this into your little booklet, and we need to explain this. Now, if we look at the graph, you can see that the body's response to the primary infection. So here, if we are injected with the antigen, or we're exposed naturally by getting a cold, then the body has been exposed to the antigen, either through injection, as in vaccination, or through disease. Now, it may take a week, week and a half before the body starts to produce antibodies. So there's what we call a latent period, a period where there seems to be no action. And then all of a sudden, the concentration of antibodies starts to increase after a week and a half of exposure. Okay, but then as the body overcomes that infection, that antibody production declines. Now, if you're reintroduced within 4 weeks, such as, again, a booster injection after 4 weeks, then the body has now kind of like produced those memory cells. We talked about it, and that's what we need to link. It actually produces the memory cells, so that the second response, that latent period, is very short. And you need to point this out, the difference between the time difference is long in the primary latent period, but short in the secondary latent period. And this is because of your memory cells. They've got that blueprint. And on the second exposure, you produce a massive amount of antibodies. And this is the theory behind any vaccination, whether it's the flu vaccine, whether it's the COVID vaccine. If the body is exposed, then the chances are that you will produce a much better response with a booster. And that's why we've been going through these processes of booster injections. Now, the problem is that we have is that these bacteria and viruses mutate. This is why we've had, you know, the alpha COVID, we've had the Delta, the Omicron. Now we've got variants of the Omicron COVID vaccine, because the virus themselves mutate, again, like this idea of changing their membrane coat. And similarly for bacteria, they change their protein coat, and it's the proteins that acts as the antigens. So that's what the body is responding to. And if it changes it and the initial antigen has disappeared and it has a new antigen on its surface, then the antibodies won't recognize it. And so therefore, we need to kind of like have a new vaccine. This is why we're continually monitoring flu around the world. This is all in response to the outbreak of Spanish flu after the Second World War. The First World War, 1918, 1920. After that war, in that period of 1918 to 1920, there was a pandemic that killed over 40 million people. We're in a pandemic at the moment, which is a worldwide epidemic, so hence the word pandemic. And because of the exposure and this virus mutating so quickly, we need a whole new series of drugs. And that's why we have those new antiviral drugs for COVID. So again, here's the explanation. At day 0, the body is exposed, injected to the antigen, and that's the protein. After 1.5 weeks, that latent period, antibodies start to increase. And then in the as the body overcomes that particular infection, we can then have a second booster, and it's a far quicker response. And that shows that the primary response, the body's immune system, has produced these B memory cells, because in this case it's the B memory cells that are producing antibodies. Remember, T lymphocytes do not, but it does stimulate the B cells to remember to produce these antibodies, okay, and in a much larger concentration. And that, folks, is it for PowerPoint 3. Hopefully you can play this over again and again if you need to go over stuff and bring questions into class. So only one more video and PowerPoint to do in this series. So that would be PowerPoint 4, video 4, which I hope to do later in the week and get that up on the system so you can carry on at your own pace then, because we need to do this and the pharmacology before the end of June, because after that I'm retired. Okay, so we need to make sure that we get that all done. Okay, so bye for now, see you in class.